My association here with this BeeHIVE cluster is partly because we are all having a common interest, which is to deal with bees, and not just honeybees, but also native pollinators. My function is as provincial apiarist. I look after the bee interests for the province of British Columbia. While many of my colleagues have highly specialized research directions, my presentation is going to be quite different. It's going to be more on a global level. We're looking at not all these small little individual facets of bee research, but we like to put in a larger, more collective and a more worldwide view on things. And this is particularly to do and address the issue of pollinator declines worldwide, and it is due to environmental stresses that are being applied to the environment. The biggest trends that have taken place are in honeybees, and because honeybees is a form of livestock that is managed by humans, and we can mitigate the impact of negative influences, be it pesticides or habitat destruction or things of this kind. We are now facing the very edges of where bees can actually function. We can no longer continue to expand and expect more out of them than what we already do.
I'm interested in determining the causes of the decline in the population levels of the Rufous Hummingbird. It's the only species of hummingbird that's declining in its occurrence in Canada. And we're looking at a variety of stressors that might be occurring to their population, including pesticide exposure. But we're also interested in determining where that pesticide might be coming from, and that's where a crossover between the warm-blooded pollinators and the bees occurs. Bees collect nectar, they bring them back to their hives, and so we're working with other people in the BeeHIVE collective to measure pesticides in the nectar. And that nectar is consumed by both the hummingbirds and also by the bees. And so we're interested in the hummingbird health, but we're also interested in how bees might be exposed to pesticides. And in particular, we're interested in the neonicotinoid pesticides.
I work on what role native pollinators play in crop pollination. And since we've found that they actually play a very important role in crop pollination, we're interested in how we can bring them back into farming landscapes where oftentimes they no longer are very abundant or very diverse. The way that we farm is really impactful for many components of biodiversity, including our native pollinators. And our native pollinators include many different species of bees, as well as flies, wasps, and other insects. The way we farm now is very intensive. We have very large expanses of the same crop. We often use a lot of pesticides on those crops, and those two factors make these landscapes less favorable to insects that need a diverse array of floral resources and undisturbed ground in which to nest, or trees and other kinds of habitat elements in which to nest. And so we found that in these intensive agricultural landscapes, we no longer see the diversity of wild native pollinators that we used to see. And yet these wild native pollinators are extremely important and complement the managed European honeybee in providing crop pollination. And so our work is all about how can we re-diversify these agricultural landscapes, bringing in specific habitat elements like hedgerows, for example, that consist of varied plantings of different native plant species that flower at different times, or even diversifying the types and numbers of crops so that we can provide floral resources for different species. Many of the people in the BeeHIVE cluster have lots of diverse and long-term experience, including experience in the agricultural sector. So I think that through collaborations, we can craft something much more comprehensive than I'd be able to do on my own. So I think that that's one of the reasons for joining the BeeHIVE cluster.
So most of my work focuses on trying to measure the toxic chemical exposures that happen in pregnant women and children, and then follow those children out until they mature to find out whether it damages their brains, whether they have problems with ADHD or learning problems. We also try to measure the exposures that can happen and identify the sources, whether it's paint or dust or water, and give us an idea of what we can do to prevent further exposures in the future. I hope to gain a number of things from working with the BeeHIVE cluster. One is to begin to take some of the work we've done in humans and children and see how we can apply that to bees. But also what we can learn from bees that's relevant to children. Like just today we learned that the way the bees pick up lead might be very similar to the way kids pick up lead that we showed 20 years ago. And so there's a lot we can learn by looking across species. The other thing I hope to learn is more and more we recognize how interconnected we all are. That what happens to bees happens to humans. We've learned from working with children that, despite the fact that we put so much money into genetics, that our health is really a function of the environment we live in, that we grow up in. And I imagine that's true for bees as well. So I hope to learn how some of these patterns go across species, that we can learn from other scientists.
At Macquarie University, the work that I do along with my group, we look at environmental pollution, air, water, soil, dust, people, biota, and also we've done work in red wine and honey. And what we're really interested in understanding is what the contaminants have done to those various different matrices and what the risk to human health may be. I'm really interested in understanding what the impact of contaminants are on the environment and also human health. An additional aspect is, once we've understood what those contaminants are doing and where they are, is to look at solutions to solving that problem. And also assisting people to provide a solution for themselves. I think the key to making this sort of science work is to make it human level so people can interact with it on an individual basis, and then you take that and then you grow it amongst many, many people to get a population-wide study in order that people engage with the issue and then the solution.
My own research interest, some of the projects I conduct for myself is to elucidate the reaction pathways of trace elements and isotopes in natural processes. The first time when we conducted Julia's samples as a contract, which we do all the time in our lab, I was very fascinated by the idea of analyzing honey, and that goes back to my heritage. Honey was a very important ingredient or a very important substance in our culture. I am from Afghanistan originally, and so I saw together with Dominic a huge potential to understand the chemical composition, basically the secret of honey, to analyze and to have the chance with Hives for Humanity to understand more about honey and its production. And this cluster basically gives us the opportunity to really elucidate honey from different perspectives, economic perspective, cultural perspective, and biochemical perspectives.